My name is Sherry Xie. I'm a bioinformatician in the Wang lab at the Icahn School of Medicine at Mount Sinai, and I'll be demonstrating how to use the ARCHS4 resource developed by our lab to query uniformly processed RNA-seq samples as well as individual genes. From the landing page of ARCHS4, we can see an explanation of the resource and the data processing pipeline. If we scroll down, we'll also see pie charts showing the breakdown of the available samples across the different geo platforms, human tissues, and mouse tissues. Clicking on either the Visualize button at the top or on a pie chart slice will take you to the data visualization page. Here, you should see a three-dimensional t-SNE plot, with each point on the plot representing a single gene expression profile. You may use your mouse to rotate, pan, or zoom into the plot. The highlighted points represent sample gene expression profiles corresponding to the tissue or cell type we just selected, which in this case was neurons. The search result box at the bottom of the page shows the color legend for the highlighted samples, which can be modified, and we also see that there are 5,325 samples from 182 series corresponding to neurons. Clicking the R download button will download an R script pre-configured to programmatically download all highlighted samples. You can also highlight another tissue or cell line in the plot using the menu to the left. For example, we can also choose to visualize all samples corresponding to the MCF7 cell line, which are now highlighted. Clicking the delete button will remove the corresponding highlights from the visualization. You may also use the metadata search bar to add additional sample highlights based on keywords or geo-IDs. Switching to the mouse view shows a similar plot of all the mouse samples. Note that the human projections are no longer displayed, but the search results are still saved, and clicking back to the human visualization will redisplay the highlighted human samples. Following the same process as before, we can highlight mouse samples corresponding to tissue types, cell lines, or metadata terms on this plot, and download them through a customized R script. Aside from metadata terms, we can also run a signature query using the Signature tab on the left, or by selecting the Signature option from the Search drop-down menu. If we input a list of up and down genes along with a descriptive signature name, gene expression profiles from samples that are similar to the input signature will be highlighted in the plot. Similarly, the Enrichment tab, which can be accessed through the menu on the left or via the Search drop-down menu at the top, allows us to view all profiles that are enriched for a given gene set. After selecting a gene set library, users can choose from any of the available gene sets. Depending on the selected direction, the resulting highlight will show sample profiles for either the top 250 up genes or top 250 down genes based on z-score are enriched for the selected gene set. You can also manually define a cluster of samples by clicking the scissors tool in the upper right corner, then clicking and dragging over any samples you would like to select. You can also minimize or maximize the visualization at any point by clicking the double arrow icon in the top right corner or by using the drop-down menu under Visualize. In addition to samples, ARCHS4 also allows users to query genes by selecting the Gene option at the top left and either human or mouse species. In this plot, each point now represents an individual gene. For the sake of this demo, I'm going to go ahead and delete all of the previous searches. Using the drop-down menus, we can then choose genes from different gene set libraries to highlight on the plot. Clicking on the number of genes in a gene set displays links to each gene's individual page, which we will go over shortly. Clicking on the Enricher logo will show the enrichment results for the given gene set in Enricher, while the Download button automatically generates a text file containing all of the genes in the set. We can also search for individual genes, which will open the gene page in a new tab. The top of the page provides a brief description of the gene and links to individual sections on the page. For each gene, ARCHS4 predicts functional annotations, such as biological processes from gene ontology, for each gene by measuring its correlation with the known set of genes associated with a term. Terms that are already known to be associated with a gene are marked with an asterisk and highlighted in aqua. You can use the search bar to look for specific terms or keywords. Other functional predictions include upstream transcription factors, mouse and human phenotypes, kinase interactions, and pathways. This page also lists the top 100 most similar genes to the selected gene based on co-expression across the ARCHS4 database, with the option to submit the entire set of co-expressed genes to Enricher for enrichment analysis. Finally, the expression of the gene across various tissues and cell lines is plotted. Hovering over a given bar shows the median expression of the gene across samples corresponding to the tissue or cell line. These images can be exported using the button at the top right of the window. The download page provides all data files created for ARCHS4. The links at the top of the page show all available files, and the most updated data release, consisting of separate human and mouse gene level expression matrices in HDF5 format, is shown highlighted in green and labeled with the associated ARCHS4 and Ensemble versions. Older versions of the gene expression matrices and the corresponding transcript level expression files and transcript TPM files are available further down on the page. Additionally, the resource also provides other types of data, such as the t-SNE visualization coordinates, pairwise gene correlation data, gene function predictions, and more. The ARCHS4 Google Chrome extension allows users to access and download the processed gene counts and visualizations for any RNA-seq data available in ARCHS4 when they visit the GEO landing page for the original data. Finally, the help page contains detailed information on the data, files, and functionality of ARCHS4, including and beyond what is covered in this demo. In addition to video and text tutorials for using the web interface, the help page also provides explanations of the HDF5 format used for all ARCHS4 data downloads and documentation for accessing ARCHS4 through an API. The ARCHS4 project is supported by grant funding from the National Cancer Institute Informatics Technology for Cancer Research Program.